Hello everyone. In this class, we'll try to learn about the cerebellum. The learning objectives of this class would be to draw schematic diagram, depict divisions and functions of cerebellum, list the afferent inputs to cerebellum, remember the arrangement and functions of cells in the different layers of cerebellum, appreciate the internal neuronal arrangements and their functions within the cerebellum, understand the influence of deep cerebellar nuclei with descending pathways, list and describe the functions of cerebellum, list the features of cerebellar disorder, list the cerebellar function test, and describe the connection and functions of cerebellum. Cerebellum literally means little brain. Cerebellum is situated posterior to the brainstem. Cerebellum is a vital for regulation of posture and movement. It receives input almost all the sensory modalities. From the spinal cord, it receives proprioceptive inputs. It receives special sensory inputs from visual, auditory, and vestibular structures. It projects to almost all areas of brain that are involved in the motor activities. Thus, cerebellum plays critical role in motor control by integrating sensory and motor information in the brain. Therefore, cerebellum strongly influences all aspects of movements starting from rate, range, force, and direction to the termination of movement. Hence, damage to cerebellum results in severe incoordination of movement. Cerebellum directly projects into brainstem nuclei that give rise to major descending pathways. Therefore, damage to cerebellum results in severe postural abnormalities. Cerebellum also regulates vestibulocochlear reflex and motor learning. Cerebellar organization. Cerebellum is located in the posterior cranial fossa behind the brainstem. It is connected to midbrain through superior cerebellar peduncle, to pons through middle cerebellar peduncle, and to the medulla through inferior cerebellar peduncle. The surface area of cerebellum is about 75% of the cerebral cortex, but in weight, it is only 10% of the cortex. Thus, cerebellar cortical tissue is much folded. There are two main fissures in the cerebellum that divide it into two major parts. The posterolateral fissure that separates the flocculonodular lobe from the rest of the cerebellum, and the primary fissure that separates the anterior lobe from posterior lobe. If you see the functional divisions of the cerebellum, it is divided into three major subdivisions: vestibulocerebellum, spinocerebellum, and cerebellocerebellum. Vestibulocerebellum is also called as archicerebellum, as it is phylogenetically is the oldest part. It consists of flocculonodular lobe. This part of the cerebellum is called as vestibulocerebellum for its extensive and reciprocal connection with the vestibular nuclei. It is concerned with the equilibrium and learning induced changes in vestibulo-ocular reflex. Spinocerebellum. This is also called as paleocerebellum as it is intermediate in its development. It consists of vermis and paravermal regions of the cerebellum. It is called as spinocerebellum as it receives proprioceptive and other sensory inputs from all the body parts through the spinal cord. It also receives input from motor cortex where the motor planning is carried out. By comparing plan with performance, it smoothens and coordinates movement. The vermal portion of the spinocerebellum projects into the brainstem areas that control axial and proximal limb muscles. Therefore, vermal spinocerebellum controls posture. The paravermal region of spinocerebellum projects into the brainstem nuclei that influence distal limb muscles. Therefore, paravermal spinocerebellum controls skilled voluntary movements. Cerebrocerebellum is also called as neocerebellum as it is the newest phylogenetically. It consists of two main cerebellar hemispheres. It is called cerebrocerebellum for its connections with the cortex. Cortex projects to neocerebellum via pontine nuclei, hence this is also called as cortico-ponto-cerebellum. As it interacts with cortex, it is involved in planning and programming of the movements. This is the diagram showing the functional divisions of the cerebellum. There are three uh, functional divisions: vestibulocerebellum, spinocerebellum, and the cortico-cerebellum. Whereas the vestibulocerebellum is important for balance and eye movements, whereas spinocerebellum is important for the muscle tone, and whereas the neocerebellum is important for the motor planning. Coming to functional histology, cerebellum is divided into outer cortex and inner part containing deep cerebellar nuclei. Cerebral cortex has three layers. The outer molecular layer, cerebellar cortex has three layers: outer molecular layer, middle Purkinje cell layer, and inner granular layer. The molecular layer contains interneurons that are basket cells and stellate cells, and Purkinje layer is which contains Purkinje cells. Purkinje cells are the largest neurons with extensive dendritic branches. The dendrites of Purkinje enter into the molecular layer, and the axons of interneurons of the molecular layer project to the dendrites of Purkinje cells. Purkinje cells also receive input directly from the climbing fibers. Purkinje cells are only cells that project from the cortex of cerebellum to deep cerebellar nuclei. Thus, Purkinje cells are connecting links between cerebellar cortex and deep cerebellar nuclei. The granule layer contains granule cells and Golgi cells, which are usually the interneurons. The Golgi cells that project to the granule cell and modify the granular cell output. The granule cells receive input from the Mossy fibers and project to the Purkinje cells, basket cells, stellate cells, and Golgi cells via parallel fibers. So this is the histological structure which is showing the different cells, the different layers, the molecular layer, Purkinje layer, granular cells, granular layer, and the white matter. The molecular layer contains mainly the stellate cells. The Purkinje cell contains mainly the Purkinje cells, and the granular layer, they are mainly made up of the Golgi cells, basket cells, granule cells, and also they are made up of the glomerulus. Deep cerebellar nuclei. There are four deep cerebellar nuclei. Nucleus fastigius. The nucleus fastigius is present in the deep vermal portion of the cerebellum. The vermal cortical portion of the spinocerebellum projects into the fastigial nucleus. Nucleus globulus and nucleus emboliformis. The globus and emboliformis are the nuclei commonly known as nucleus interpositus. The paravermal portion of the spinocerebellum projects into nucleus interpositus. Nucleus dentatus. This is present in the hemispheric portion of the cerebellum. It receives input from neocerebellum. The name of the nucleus is dentate for its appearance, which has a teeth-like serrated morphology. The deep cerebellar nuclei project to the different parts of the brainstem and thalamus. Coming to cerebellar connections, the inputs to the cerebellum. Cerebellum receives somatosensory inputs from almost all parts of the body and inputs of all sensory modalities including special sensory inputs. The cerebellar afferents are mainly the vestibulocerebellar tract. Through this tract, the cerebellum receives impulses directly from the vestibular apparatus and also from the vestibular nuclei. Dorsal spinocerebellar tract. This tract conveys proprioceptive and exteroceptive impulses from different parts of the body to the cerebellum. Ventral spinocerebellar tract. This pathway also conveys proprioceptive and exteroceptive impulses from different parts of the body. The cuneocerebellar tract. This tract originates from lateral cuneate nucleus in the caudal medulla and conveys proprioceptive inputs from head and neck. Whereas tractocerebellar tract, it conveys the visual information from superior colliculus and auditory information from the inferior colliculus to the cerebellum. Pontocerebellar tract impulses which from the motor cortex reach the cerebellum via the pontine nuclei. Olivocerebellar tract, the proprioceptive inputs from the whole body reaches the cerebellum via inferior olive. The inferior olivary nucleus is located in the rostral medulla that receives input from the vestibular system, spinal cord and cerebral cortex. It projects the cerebellum via climbing fibers. Modes of input to the cerebellum reach via three routes, that is Mossy fibers, climbing fibers and other inputs. The Mossy fibers are the major source of inputs to cerebellum. These fibers carry direct proprioceptive inputs from all parts of the body and also convey input from cerebral cortex. Mossy fibers project mainly into the granule cells. The climbing fibers convey inputs from inferior olivary nucleus to cerebellum. Inferior olivary receives proprioceptive inputs from all parts of the body, and climbing fibers project to Purkinje cell of cerebellum. Other inputs, the cerebellum receives monoaminergic inputs and inputs from the thalamus and other parts of the brain. These fibers project to the deep cerebellar nuclei. So these are the different cerebellar inputs through the climbing fibers, through the Mossy fibers and other system. The tracts involved for the climbing fibers are olivospinal tract, and they bring proprioceptive inputs from whole body via relay in the inferior olivary nucleus. Whereas Mossy fibers receive information from dorsal spinocerebellar tract, ventral spinocerebellar tract, vestibulocerebellar tract, tectocerebellar tract, cuneocerebellar tract, cortico-ponto-cerebellar tract, and these are going to receive information, the proprioceptive information from the different parts of the body, vestibular apparatus and the eye and head neck. The other system like monoaminergic outputs, that is serotonergic, noradrenergic fibers, they receive from the raphe and raphe magnus and locus coeruleus respectively, and the thalamus is going to receive information from the brain and other structures. The cerebellar outputs, the different parts of the cerebellum project to various descending pathways via deep cerebellar nuclei. Deep cerebellar nuclei are output pathway for the cerebellum. The output from vestibulocerebellum. Vestibulocerebellum directly projects to the vestibular nuclei without any relay in the deep cerebellar nuclei. Thus, vestibulocerebellum directly controls vestibulospinal tract. The output from the spinocerebellum. The vermal portion of the spinocerebellum projects into fastigeal nucleus, which in turn projects to pontine reticular formation and vestibular nuclei in the brain stem. Thus, the vermal part of spinocerebellum controls the activity of pontine, reticulospinal and vestibulospinal tract. The paravermal portion of the spinocerebellum projects to the nucleus interpositus, which in turn projects into red nucleus. This paravermal part of the spinocerebellum controls the activity of cerebrospinal tract. The output from cerebrocerebellum. The cerebellar hemisphere projects to the dentate nucleus, which in turn projects to the motor cortex via thalamus. The cerebrocerebellum controls the activity of the cortico-spinal tract. As different parts of the cerebellum project into all the motor nuclei in the brainstem and to the motor cortex, cerebellum controls the activities of all descending pathways, that is corticospinal as well as extrapyramidal system. That is why the disease of the cerebellum affects both regulation of posture and skilled voluntary movements. So these are the different tracts which are going to run into the different peduncles, that is superior, middle and inferior peduncles. In superior peduncle, the cerebrotectal-dentato-thalamic and dentatorubral cerebello-tegmental pathways they are conveyed, whereas the pontocerebellar pathway run in the middle uh, cerebellar peduncle, whereas through the inferior peduncle uh, they convey cerebello-reticular, vestibulocerebellar, olivocerebellar, archeocerebellar, trigeminocerebellar, cerebrospinal, and spinocerebellar tracts. Internal connections of the cerebellum. The cerebellum receives inputs from two sources, that is climbing fibers, that is from the olivary nucleus and the Mossy fibers. Purkinje cells are stimulated directly by climbing fiber input, whereas Mossy fibers stimulate Purkinje cells indirectly via granule cell parallel fiber pathways. The Mossy fibers project to the granule cells, and granule cells via its parallel fibers provide excitatory input to the basket and stellate cell and Purkinje cells. The basket and stellate cells that are activated by Mossy fiber parallel fiber pathway finally inhibit Purkinje cells. So this is an example for feed-forward inhibition. Whereas granule cells also stimulate the Golgi cells, the interneurons in the granular cell layer, so which in turn inhibit the activity of granule cells. This is an example of local feedback inhibition, and it is meant to regulate the granule cell output. So this diagram showing the internal connections. Okay, so where the Purkinje cells is going to receive information from the climbing fibers and by the Mossy fibers through the granular cells. Okay. The climbing fibers, they are mainly the excitatory to the Purkinje fibers, whereas Mossy fibers through the granule cells they are going to excite the climbing fibers. And the granule cells they also in turn stimulate the stellate cells and the basket cells, which are going to in turn inhibit the Purkinje cells, which is called as feed forward inhibition. Okay. So that is stimulation of the granule cells. They are going to stimulate the stellate cells and basket cells. In turn, they are going to inhibit the activity in the Purkinje cells, which is called as feed forward inhibition. Whereas the granule cells, which in turn stimulate the Golgi cells, in turn they're going to stimulate or rather inhibit the granular cell activity, which is called as local feedback inhibition. And the Purkinje cells ultimately they are going to inhibit the deep cerebellar nuclei. The Purkinje cells ultimately they tries to keep check the deep cerebellar nuclei. They try to inhibit the deep cerebellar nuclei, but the output from the deep cerebellar nuclei is always excitatory, as it is going to be influenced by the other inputs from the different parts of the body, that is through the Mossy fibers or through the climbing fibers directly. That one should remember. The excitatory output from cerebellum. Purkinje cell output to the deep cerebellar nuclei is inhibitory because the neurotransmitter secreted by Purkinje cell is GABA. However, deep cerebellar nuclei receive excitatory inputs from Mossy fibers and climbing fibers and from the other sources. Therefore, in spite of inhibition by the Purkinje cells, the output of the deep cerebellar nuclei to the brainstem is always excitatory, as we discussed. The internal circuitry of cerebellar neurons is designed mainly to modulate the excitatory output of the deep cerebellar nuclei. Therefore, the lesion of the cerebellum in human being results in hypotonia. So this is the diagram showing the effect of the cerebellar cortex on the Purkinje cells. The Purkinje cells, they're going to they're going to affect the different deep nuclei. Okay, so which were from the cerebellum, usually from the Purkinje cells, there will be negative influence over these nuclei, but because of the other inputs, the cerebellar nuclei, they are always excitatory to the brainstem nuclei. So they have excitatory effect on the red nucleus, vestibular nucleus, and the brainstem nuclei, and thus they are going to increase the muscle tone through different pathways, that is reticulospinal, vestibulospinal, cerebrospinal, and also through the pyramidal tracts. The dentate nucleus, it has its connection to the thalamus, and thalamus is going to modulate the activity of the cortex, and the corticospinal tract to the descending pathway, they are going to control the muscle tone. Purkinje cell activity. Purkinje cells exhibit two types of action potential, that is the simple spikes and complex spikes. Simple spikes action potential is generated in response to stimulation of Mossy fiber parallel fiber input. The complex fibers, that is multi-peak spike action potential, is generated in response to stimulation of the climbing fiber input that comes from olivary nucleus. These complex spike action potential are involved in motor learning, as climbing fiber activity is observed to be increased when a new motor task is learned. They also produce long-term adjustment in motor responses. Coming to the functions of cerebellum, the cerebellum controls the postural balance and equilibrium. This is the function of vestibulocerebellum, which has extensive reciprocal connection with the vestibular nuclei. Afferents from vestibular apparatus in the inner ear project to the vestibulocerebellum via vestibular nuclei. The vestibulo-ocular reflex. The vestibulocerebellum is concerned with learning-induced changes in vestibulo-ocular reflex. Cerebellum is important for smoothing and coordination of the movement. This is the function of spinocerebellum that receives the proprioceptive and other sensory inputs from all body parts through spinal cord. It also receives input from the motor cortex, where the motor planning is carried out. By comparing plan with performance, it smoothens and coordinates the movement. It is important for control of posture. The vermal portion of the spinocerebellum projects into brainstem areas that control axial and proximal limb muscles. Therefore, vermal spinocerebellum has a profound influence on posture. Cerebellum is important for control of skilled voluntary movements. The paravermal region of the spinocerebellum projects into the brainstem nuclei that influence the distal limb muscles. Therefore, paravermal spinocerebellum controls skilled voluntary movements. Cerebellum controls all aspects of movement, starting from rate, range, force, and direction to the termination of the movement. Although functionally, cerebellum has three lobes, that is vestibulospinocerebellum, vestibulospino and neocerebellum, they work in a coordinated manner. That means it acts as a comparator of a servo mechanism. The cerebellum receives information from corticospinal output transmitted to the muscles, receive proprioceptive inputs from the muscles via spinocerebellar tracts that informs about the ongoing movements and position of the limbs, and also receives all special sensory inputs, that is visual, auditory, and from the vestibular inputs. Cerebellum projects to cortex via red nucleus and pontine nuclei. The cerebellum coordinates all the cortical and spinal information and appropriately modifies the ongoing movement via its influence on all descending pathways. So it sends error signals to the cortex for alteration in the programming of the movement of any desirable change in the motor output to be achieved. It is also important for planning and programming of the movements. So this is the function of neocerebellum that interacts with the cortex. And hence, the neocerebellum controls planning and programming of the movements. This is the diagram showing how the cerebellum is going to control the activity of the motor cortex, and how it is going to affect the brainstem nuclei, that is anterior horn cells. It's also useful in the control of muscle tone and stretch reflexes. Cerebellum influences the activity of major descending medial system pathways through its output from fastigeal nucleus, especially the vestibulospinal and reticulospinal tract. As the vestibulospinal tract mainly controls alpha motor neuron activity and reticulospinal tract controls the gamma motor neuron activity, in the spinal cord, the cerebellum is one of the major sites of alpha-gamma co-linkage. In human beings, output of the deep cerebellar nuclei to the brainstem motor nuclei is excitatory that facilitates the muscle tone. Therefore, cerebellar disorder produces hypotonia. Though cerebellum has profound influence on all descending brainstem pathways, its influence on stretch reflexes is minimal, except in some patients it is pendular in whom the knee jerk is observed, the pendular knee jerk. Therefore, the stretch reflexes remain usually normal in cerebellar disorder. However, in animals, the connection with cerebellum with the brainstem nuclei is complex, which inhibits the output from inhibitory reticular area. Therefore, cerebellectomy in a decerebrated animal increases the spasticity of extensor group of muscles. That is, it is going to facilitate the decerebrate rigidity. Then they are important for the control of movements of one side of the body. The motor cortex, one of the side is connected to the cerebral hemisphere of the opposite side through the closed feedback circuit, which is known as cerebral cerebral circuit via the cortico-ponto-dentato-thalamo-cortical connections. Thus, each cerebral hemisphere influences the output of opposite cerebral cortex. However, cerebral cortex via cortical tract that decussate to the opposite side just after passing through the pyramid controls the motor function of contralateral half the body. Therefore, due to double decussation, each cerebral hemisphere controls the movement of its own side of the body. Learning and improvement of the motor skills. Cerebellum plays a critical role in comparing information of the ongoing movements and changes required to improve the performance. Hence, for every activity, cerebellum improves learning and performance. Therefore, also controls the long-term adjustment of motor skills. Especially the climbing fiber inputs produce complex spikes in Purkinje cells that is involved in motor learning. Climbing fiber activity is increased every time a new activity is learned. They are also useful in eyeball movement. The paraflocculus and pyramis of cerebellum are concerned with movements of eyeball, especially in upward direction. Stimulation of these parts of cerebellum causes upward eye movement of the ipsilateral side, especially the visual judgment of distance is function of cerebellum, which is more developed in monkeys. Important for vestibular function. For its dense and reciprocal connection with vestibular reflex, vestibulocerebellum is involved in the control of all vestibular functions such as balance during movement, execution of vestibulo-ocular reflex, vestibular postural reflex, and change in the body posture and movement in response to head movement and acceleration. Coming to cerebellar disorders, diseases affecting flocculonodular lobe results in abnormalities of maintaining equilibrium. For example, stimulation of vestibulocerebellum and vestibular nuclei leads to motion sickness. Intractable motion sickness, in fact, is cured by selective removal of flocculonodular lobe. Features of cerebellar disorders. They are going to depend on the part of cerebellum affected and whether the cortex or the deep cerebellar nuclei are involved in the disease process. Effect of lesion of one side of cerebellar hemisphere manifest on the ipsilateral side of the body. In general, cerebellar disorders have following features. There won't be any paralysis, that is, the voluntary movements are intact, though defective. Usually reflexes are normal, except that sometimes the pendular knee jerk is elicited. There won't be sensory deficits. Hypotonia is a usual feature. There will be ataxia. Motor deficits in the cerebral disorder manifests mainly in the form of ataxia, which is defined as the defect in coordination due to the errors in the rate, range, force, and direction of movement. If any cerebral cortex is involved in the disease process, ataxia is temporary. But if the lesion involves deep cerebellar nuclei, the ataxia almost becomes permanent. Ataxia can manifest in the following form. It could be a drunken gait, scanning speech, or dysmetria. Drunken gait is unsteady and wide-based gait, whereas scanning speech is ataxia involving the muscles of speech uh, manifest in the form of scanning speech. The patient scans the syllables while speaking. So there can be dysmetria when the patient attempts to touch any object. Usually the hand overshoots instead of reaching the target, and this is called dysmetria, that is inability to measure the length of or the distance. So this is also called as pass pointing. So there can be intentional tremors due to the dysmetria. The corrective measures are immediately initiated, but this time hand overshoots in the opposite direction. Repeated overshoot and recorrection results in intentional tremor, that is hand oscillation back and forth. The tremor is not seen at rest. So there can be rebound phenomena. This results due to inability to put on brake or suddenly stop on ongoing movement. For example, if the patient is asked to flex his limb against the resistance and then asked to stop immediately by withdrawing uh, the resistance, he cannot stop. Rather, the arm moves with the wide arc. So this is called as rebound phenomenon. There can be eudiacokinesia. Eudiacokinesia, inability to perform alternate movements rapidly, called eudiacokinesia. For example, a patient cannot perform supination and pronation rapidly. So there can be decomposition of the movement, inability to perform movement that involves more than one joint simultaneously. Therefore, cerebellar patients dissects such complex movement and performs movement at each joint slowly and separately. So there can be inability to carry out long-term adjustment in motor response. So there can be defect in vestibulo-ocular reflex leads to pathological nystagmus. There can be Charcot-Strand presence of nystagmus, intentional tremors, and scanning speech, or lalling speech like baby, so which is seen in cerebellar disorder, and disseminated sclerosis that affects the cerebellar functions. Friedreich's ataxia is a form of hereditary ataxia in which spinocerebellar tract degenerates, producing the ataxia as described above. Some of the cerebellar function tests. Many clinical tests detect cerebellar functions. These are tests for coordination in upper limbs and lower limbs. For upper limbs, it is finger nose test, making circle in the air, and in the lower limbs, it is knee heel test, walking on a straight line. And to test the postural stability, uh, you're supposed to ask to stand erect with the feet closed but eyes open. Then assessment of various aspects of ataxia by eliciting different movements as described above. And you have to assess the gait and the speech. This also becomes part of cerebellar function test. So this is in brief about the cerebellum, its connection, functions, and the disorder of the cerebellum. Thank you.